Have you ever wondered how your metabolism works during overnight fasting? Our body's metabolism continues during the night since the functioning of organs, such as the heart and the brain, among many other processes, requires a source of energy. After several hours of fasting, blood glucose levels decrease. Maintaining blood glucose levels is essential during fasting since it constitutes the main source of energy for the brain and the only source used by red blood cells. The nervous system, testes, kidney medulla and embryonic tissues also use glucose as their main energy source. The decrease in blood glucose levels is sensed by the alpha cells of the pancreatic islets of Langerhans, which release the hormone glucagon under these conditions. The entry of glucose into alpha cells and the consequent production of energy is due to the activity of the glucose transporter 1. When the blood glucose levels drop, the intracellular concentration of ATP in cells decreases. This leads to the closure of ATP-sensitive potassium channels, which reduces potassium efflux. The resulting depolarization of the plasma membrane prompts the opening of voltage-dependent calcium channels. Increased cytosolic calcium finally triggers the release of the hormone into the bloodstream. Once released into the blood, glucagon coordinates metabolism in various tissues to fulfill our energy needs. The binding of glucagon to membrane receptors on different cell types triggers the activation of the enzyme adenylate cyclase, which transforms ATP into cyclic AMP. This triggers activation of protein kinase A, thereby starting a signaling cascade. As a result, the metabolism of various tissues and organs is reprogrammed. The liver plays an essential role in maintaining glucose levels under these conditions, thanks to its metabolic flexibility. As a result, liver metabolism adjusts to maintain glucose availability. When blood glucose levels drop during fasting, glucagon stimulates the release of glucose from the liver, firstly by activating the breakdown of liver glycogen. This polysaccharide, accumulated in limited amounts, is broken down to release glucose into the blood. This restores the blood glucose levels. Activation of hepatic glycogen degradation occurs via activation of protein kinase by glucagon. This causes phosphorylation and activation of phosphorylase kinase, which in turn activates glycogen phosphorylase. This enzyme is responsible for the phosphorylatic cleavage of the glycosidic bond, thereby generating glucose 6-phosphate. The ability of the liver to rapidly mobilize its sugar reserves is essential in the response to hypoglycemia in fasting. Once glycogen stores are depleted, the liver continues to produce glucose through gluconeogenesis from substrates such as amino acid generated by muscle protein breakdown, from glicerol released in the mobilization of fat from adipose tissue, and from oxaloacetate from the Krebs cycle. Glucagon triggers the activation of gluconeogenesis. Binding of the hormone to its receptors activates protein kinase A, which promotes the activation of fructose 1,6-biphosphatase, the main regulatory enzyme of gluconeogenesis, by decreasing the levels of fructose 2,6-biphosphate. Increased levels of the enzyme phosphoenol pyruvate carboxykinase, due to the induction of glucagon expression, contribute to the increase in gluconeogenesis. To maintain energy homeostasis during fasting, when energy availability is limited, other mechanisms of energy production are activated. During fasting, in particular, glucagon promotes the mobilization of fat from adipose tissue. This occurs via phosphorylation by protein kinase of the protein surrounding the fat droplet in adipocytes called perilipin, and the activation of hormone-sensitive lipase, which facilitates the access of lipases to the accumulated fat and releases fatty acids and glycerol into the bloodstream. Under these conditions of low energy availability, the use of fatty acids as an energy source is favoured. Fatty acid breakdown in muscles provides energy in the form of ATP. As the fasting time increases, the liver uses a higher proportion of fatty acids for the generation of ketone bodies. In fact, ketogenesis is also stimulated by glucagon. Ketone bodies generated from acetyl-CoA in the liver, acetoacetate, and B-hydroxybutyrate can be used by the cardiac muscle and adrenal cortex as a source of energy, and also by the brain in prolonged fasting of more than 24 hours. The functioning of the liver under these conditions is referred to as glucogenic liver, since it focuses on the generation of glucose for the brain. Liver glycogen, glycerol derived from fat mobilization in adipose tissue, and amino acids derived from protein degradation are used as substrates for this purpose. Furthermore, during prolonged fasting, ketone bodies are synthesized from accumulated fats. However, their accumulation in excess in the blood and urine is toxic to the body. It is therefore necessary for our survival to maintain a diet that ensures adequate levels of ketone bodies during fasting in order to prevent overproduction.